wanted to go over the diagram with you guys that's in your outlines, because I would normally go over this in class, but we're not in class. So let's just take a look at this. You would want to be able to recognize this picture as a cell in mitosis, and you want to be able to identify the phases of the cells by looking at what's going on inside of them. And then you want to also recognize the parts within the structures inside the cells. So let's start with just the phases. So this is a little confusing. The black arrows just refer to the circular or cyclical nature of this process. So I want to start here with this cell. So what you see in this cell is you see a nice nucleus, right? It's bound by this dark black line. The structure that you see here in yellow is going to be the chromatin, the DNA. The dark spot inside of our nucleus, right here, where it's number 7, that's going to be the nucleolus, right? So remember, the nucleolus is a dark staining circular structure inside of the nucleus, and that's visible during this phase. We also see that we have our organelle with our 2 little cylinders at perpendicular angles, so that's going to be our centrosome. Now, what phase are we looking at? Because we can see the nuclear envelope is completely intact, and because the DNA is in chromatin form, we can say that this is interphase. Now, we know that we have interphase. As we move on to the next phase, we can see what's going on here. In this cell, we see the centrosomes pulling apart from each other. We see the spindle fibers being created. The nuclear envelope, which is number 10 here, is no longer as dark, and we have our chromosome, right? Number 9 would be a chromosome, or you could say for number 9, a sister chromatid. So what phase is A? A is going to be prophase. So as we move over to the next cell, phase B, we can see that the chromosomes have been aligned along an equatorial plate. This is a hallmark of metaphase, so B is metaphase. Number 2 is pointing to these lines, right? So these are going to be our spindle fibers that are helping to push our chromosomes into that metaphase plate. And that dot in the center here of our chromosomes, that's going to represent our centromere. Okay, so we have metaphase in letter B. The next phase is going to be anaphase. So letter C is going to be anaphase. In anaphase, we can see the sister chromatids are being pulled apart. They're being pulled apart because the spindle fiber that is attached to each centromere is shortening on either side, and there is pulling those sister chromatids apart. So this is going to be number 3. We're pointing at 1 chromosome on either side, so we can call these sister chromatids, right? Because we want to say that in anaphase of mitosis, sister chromatids are pulled apart. Then our last phase, D, will be telophase, accompanied by cytokinesis. So what you can see is number 4, these are 2 new cells. We can see that our cell, the nuclear envelope, is forming again around our 2 new nuclei. And we can see that each cell will eventually have 1 centrosome, and the spindle fibers are coming back to the short form, right? They're shortening. And what's happening here, where the cytoplasm divides, is going to be called cytokinesis. All right, so that's just a quick review of mitosis.